So I'm going to talk about the genetics of neurodevelopmental disorders. It looks like I can do stuff with this. I'm sure there's a laser. Oh, okay. Maybe. Okay, so just to kind of give you the ground rules here, I'm going to use the term NDDs, or neurodevelopmental disorders, to include autism, intellectual disability, epilepsy, ADHD, OCD and tics, language delay, learning disability, dyslexia, and so on. Not all of these are at the same point in terms of new approaches, but certainly the first 3 or 4, and primarily the first 3, there's been a revolution in how we understand the risk architecture that could lead to novel ways to treat. So to kind of make that point, the first point I want to make is that this is just from, there are many papers like this. This is the one I chose that I found rather, the one that came to the very top of my Google search. But that middle column there of heritability estimates is the best guess of how much of a disorder is controlled or by genetics, right? And what you can say is PDD stands for pervasive developmental disorders. It includes autism, Asperger's, PDDs, and so on. Conduct disorder is a behavioral disorder, OCD, anxiety, ADHD, schizophrenia, bipolar, major depression. What you can see is in every example here, heritability is significant. In other words, the amount of contribution to the disorder by genes is 30, 50, 60, 90%. And it doesn't really matter if it's 30 or 80, because either way, you can use that genetic information to think about novel approaches. And you'll see that the PDDs, autism spectrum disorder, are some of the most heritable disorders we know today. All right. So if I convince you, or that paper convinced you, that there's a lot of genetics behind these disorders, then we have to try and understand what does genetics look like in this case. And one of the useful things we do is we think about genetics in 2 dimensions. One is we think about how common a genetic change is. Okay, so that's on the bottom of the scale here. Do we have a pointer, if anybody? Thanks. So there are plenty of genetic changes that are common in the population, right? And there are some genetic changes that only a few people, maybe only one person in the planet today may carry. At the same time, the second dimension we think about, which one? I don't see it. Maybe it's there. So when we think about each genetic variant, it can have a major effect, a high effect, or it can have a very low effect. And that turns out to be important, right? So those 2 dimensions kind of classify how we think about genetic architecture, and also tell us how we can go forward, what we might find and what we might do with it. So just to kind of make the point here, if we're over here, something that's both very rare and has a tiny effect is not somewhere it's not useful, right? We're never going to find it if it's both very rare and tiny effect. Things that are up here, okay, that are both common and have very high effect size, you're not going to see in a neurodevelopmental disorder. Because if they really are major risk factors for NDDs and they're common, we'd all have them, right? So what actually happens is when events like this that are actually high risk happen, they tend to go away very quickly in the human population. It's called purifying selection. That's what Darwin talked about a lot, right? And so the areas that we can actually find things, or are likely to find things, are on this kind of middle path here. Things that are rare but have high risk, and things that are common but have very low risk, right? Because anything that's rare, anything that's high risk is going to be pushed down into very rare events. Does that make sense? All right. So starting with, so let me take those same NDDs I talked about earlier and kind of give you a snapshot of where we think we are with them. We're quite certain at this point, and I'll show you data, that autism, intellectual disability, and epilepsy have a lot of these rare variants of major effect. So mutations, basically. What do you consider a mutation? We also know that those 3, as well as ADHD and OCD, have intermediate mutations of intermediate effect. So they don't guarantee one thing or the other, but they're reasonably high-risk genes. And then way down here all of the disorders and language delay, learning disability, dyslexia, and so on have a plethora of common variants of very, very low effect. So this is what's called polygenic risk. Many, many factors come together to create the ultimate phenotype. Okay. So those are kind of just to map the things I'm talking about onto this scale. And this is actually a formal study that was done in Sweden where we have a lot of national information, registry information, and clinical information. And we can show just for autism, okay, that common variation accounts for about half of the total risk. So this bit over here accounts for about half of the total risk for autism, these common variants of weak effect. In this slice over here, there's about another 10% that are these rarer mutations of major effect. Okay, so we can actually look at how these two things interact. So autism and probably all the NDDs are a combination of rare events that are very powerful and a large number of common variants that have tiny effect sizes. All right, so let's start with what's up here, where really most of the action has been. We're looking at rare genetic changes that are Mendelian. Mendel, the guy with the P's, what he discovered were things that were major effect genes, right? So Mendelian disorders are disorders where the mutation accounts for virtually all of the risk for the disorder. So within autism, intellectual disability, and epilepsy, we now are beginning to find lots of mutations just like that account in a single person for much of their risk. And so this is the results that's now online in Cell, the journal Cell. This is a study that me and my colleagues have done, a group called the Autism Sequencing Consortium, has DNA samples from 36,000 individuals and interrogates virtually every gene in the human genome in every one of those people. We have about 20,000 distinct genes in the human genome. Our current technology can easily track about 19,000 of them, so we're missing a few. But this is the summary of the findings from 36,000 DNA samples, 36,000 individuals. And the way it's mapped out is that on the bottom here, these numbers are chromosomes. Human chromosome 1, 2, 3, 4, 5, all the way up to 22. Chromosome X, Y are not on this slide, but they're sometimes called 23 or X or Y. And what's on this axis, going up and down, is the evidence. How strong is the evidence that a particular gene is associated with autism? And those little 4 and 5 letter, I did not mean to do that, 4 and 5, okay. Those 4 and 5 letter things there next to each point are gene names. So gene names, there are 20,000 of them. You can imagine they're not the most exciting names typically. Some of them are generated by computer. Oh, perfect. Some of them are generated by computer. If you don't know what it is, it's often called an ORF, an open reading frame, which is a very exciting name. But some of the genes have, this is a sodium channel, this is a structural protein, this is I won't go into all of them. But everything, everything above this red dotted line on the bottom is actually significant. Real association with risk. There are 102 of them on this map alone. So we already have, let us say, at least 100 genes, and then there's actually many more on the X chromosome, that have high risk for autism, all right? And, the ones that are at the very, very top, maybe this top 15 or so, the mutations are very, very penetrant, which means they account for most of the risk in that individual, and they're not that uncommon. None of these are common mutations, but if you look at autism, most of the genes on the top here will be found in about 1% of the individuals with autism. So 1% sounds like a tiny bit, but it's not so bad when you think about what we now call precision medicine, where you want to, like in cancer, you don't treat cancer so much as you treat the specific kind of cancer in the ideal world, right? And a lot of people think that, and generally in medicine, we're going to have to move to a model where we don't treat, we can also treat general autism as an example, but if a person's carrying a specific mutation, there may be an additional treatment that's specific for them. All right, so remember all these gene names, if you please, because they're going to come up again. Just kidding. FOXP1, ADMP, SHANK3, I'll show you on the next slide, SYNGAP1, they keep on coming up. So this is autism. If you now go to another study that was done in the UK called the Deciphering Developmental Disability Study, where they serially collected 12,000 individuals, young kids, babies sometimes, who came to a clinical geneticist because they had developmental delay. They weren't developing on the proper trajectory, either motor-wise or language-wise, or they had congenital anomalies. They took 12,000 kids and their parents, and they did exactly what we're doing in the ASC now. So this is developmental delay, intellectual disability, and the same exact structure. On the bottom are the chromosomes, 1 to 22, and here they have the X chromosome. And on this axis over here, these are huge. Minus 70 is the strongest findings you'll ever find in science. Okay? Just trust me on this. In my entire life, there's only a couple of examples like this, right? So these are unambiguously major genes for developmental delay, including intellectual disability. And I mentioned ADMP. I'm sure you remember that one. SYNGAP1. Not too many of that I mentioned previously, but they're all there. ARID1B, if I go back, right there. So there's a lot of overlap. So it's not just that these genes increase risk for autism, but also for intellectual disability and global developmental delay. They have the X chromosome on their study. Here is DDX3X, that's an X chromosome gene, now also significantly associated with autism as well. And interestingly, because it's on the X chromosome, it's specifically found almost exclusively in girls. So it's one of our first entrees into female, if you will, autism, an understudied area. And then if you look at the epilepsies, severe epilepsies, childhood onset epilepsies, and you do exactly the same thing, this is now with 17,000 individuals, they're hoping to get to 25,000 very soon. They plotted a little bit differently, but all these genes are highly significant, and these are different kinds of epilepsy. This is epileptic encephalopathy, generalized epilepsy, and non-acquired focal epilepsy. It's a little bit blurry, at least to me, but trust me when I say that some of the genes overlap. SNC2A, they're there. So for all three of these disorders, we have dozens to hundreds of genes of major effect. And as I said, they often overlap. Right? So, and this is kind of to make the point that even when they overlap, there may be some subtleties. So this is looking at all of the data from the Autism Sequencing Consortium. So this is in autism, all of the data from the DDD, so it's intellectual disability. And these are all the genes that overlap. And some of them are more likely associated with intellectual disability, and some of them are more likely associated with autism. But for the most part, to a first approximation, there's enormous overlap between these genes. So that's kind of intriguing, maybe a little hard to explain. But also from the point of view of interventions, if you develop a treatment for this gene over here, KMT5B, you almost don't care. You care a little bit. But you care less about what exactly is the presentation. Because you know if there's some biology that's dysregulated, it'll help whether it's epilepsy, autism, or developmental delay, or intellectual disability. The curious part is, why would it be that the same mutation presents this way or that way in different people? And one hypothesis is that everything else, that common variation we talked about that accounts for 50% of the risk, maybe that sculpts the final phenotype. But you need that severe first hit to even push you over the threshold at all, and what actually manifests might be tweaked, if you will, by these common variants. So let me jump down to common variants briefly. As I said, they're implicated in everything, and everything you think about in terms of human beings, it's common variation, height, IQ, hair color. All those things are lots and lots of common variants working together. But it also turns out that some of the disorders we look at are also lots of common variants working together. So this is kind of the same plot. This is for ADHD, and I chose ADHD to show that we're kind of going the same place with that as well. And again, here's the chromosomes, here's the evidence. Now you don't see too many things with a 70 or a 60 or a 50 because the sample sizes are small. As I said, these things have very weak effects, so you need a lot of data to convince yourself. But there are definitely, each one of these little pentagons, well, not pentagons, but rhomboid, what is it? It's a diamond, whatever, which goes above the dotted line, are significant. And they don't directly implicate a gene so much as they implicate a region in the genome. And these are some of those genes. right? So in ADHD and in autism and in intellectual disability, we're getting a handle on common variation as part of our understanding of risk architecture. And one of the things, and just taking that same exact data, so now you're taking all of these thousands of variants of tiny effect and asking, what do they do in aggregate, okay? And what you can see is something called polygenic risk score. Take any individual that you want, add up all of their risk score, these little tiny variants of minor effect, and ask, how do they map? These individuals over here have the lowest decile of risk, and these individuals have the highest decile of risk. And what you can see is risk for ADHD goes from a baseline of 1 to about 5-fold higher depending on the polygenic risk. All right, so in aggregate, all of those things together contribute to risk, but each one, there are thousands under this. There are thousands of variants under each one of these peaks. Each one by itself does not make any difference whatsoever, in spite of what you might hear from some of your consumer genetic testing laboratories. So, and then one of the interesting things about all of these common variants is they also speak directly to the rest, to typical development, right? So if you take the polygenic risk for ADHD, it is negatively correlated with childhood IQ, education, college completion, human intelligence, getting a college degree, positively correlated with neuroticism, depression, and so on and so forth. Some of this wouldn't surprise you. If you have ADHD or high risk for ADHD, you're going to have a hard time making it through school, right? But these kinds of associations now can be applied in the general population. And some of them are quite interesting. Polygenic risk for autism is associated with higher educational attainment, right? So whatever makes all those things coming together on the common variant space, whatever that is, actually in the general population is a very positive thing, if you like more schooling like I do. So we're beginning to learn also about, just the universe of human variation, if you will. All right, so back to this. As I said, 50% of autism is common variation. Another 10% is rare variation. We can do a lot more with these rare variants than we can with these common variants. And also, as I said, not only do we have more technology and more entree into them, but it's also true that if an individual is carrying a rare variant like this, that's something you want to address, right? Very clearly, on an individual level, that's where you want to go. So, just to be clear, oops, I see what I was doing here. This is the problem with getting your slides in on time. I forget them. So I said about rare variation in autism. This is the same exact thing now with common variation, just came out. Again, the chromosome, and there are just an emerging common variant loci in autism. And just like I said before, if you look at the common variation for autism, it's associated with higher IQ, higher educational attainment, more likely to be in college, and so on and so forth. So these are actually positive things, right? So, how do we take advantage of the positives and push away or eliminate this negative major mutation that some of these kids are carrying? And this is in the general population. I maybe won't go into it, but basically saying the same thing. You take the polygenic risk for autism, and you actually see in the general population changes in sociability that kind of go along with risk. All right, so back to these rare variants of major effect. Properly, by the time they're really, really powerful genetic changes, we can call them mutations. A mutation is something where you kind of say, if you have this change, you're going to likely have a deleterious outcome. So I'll just summarize, and I have a bunch of slides to make it kind of clearer to the real experts in the room. But one thing that's very clear from those first 100 genes is that it's not a single pathway. There will not be a single drug that treats all of these different disorders equally well, different gene mutations equally well. That's why precision medicine is critical. And cancer is a great example. There are cancer therapeutics that just kill any cancer cell. They also kill healthy cells too. That's the side effect part. But as you probably know, and I'm sure most of you have somebody in your family that might be taking advantage of the new magic bullets that are specifically targeting a kind of cancer, right? And so in autism, intellectual disability, epilepsies, we're probably going to have to go there. but it's not going to be one magic bullet. Right? It's going to be different bullets for different kinds, different biotypes. All right. What we're finding to date, and it's partly because of the way these things happen, they're overwhelmingly dominant mutations, which means that there is a dominant mutation is where everybody has 2 copies of every gene, almost every gene. A dominant mutation is where the mutation on one of them causes the manifestation. You don't need a mutation in both. What that means is every time you see a mutation like the ones I've showed you, there is a second copy of the gene that's not mutated. So if you could take advantage of that copy and make it increase its levels or something like that, you have a way to actually help in a kind of fairly directed way. They're primarily loss of function, which means that most of the time it's a mutation that stops the gene from doing its proper work. It's often a U-shaped curve for the aficionados. It means that too much or too little can be deleterious, so you have to think about how much you want to put back in. You don't want to go overshoot it too much. These tend to be, the ones I'm showing you, all those genes tend to be very severe, so the unmet need here is huge, right? These families are crying for help. And one of the things that I think our biotech and pharma partners want to know is, okay, you have a mutation that happened before conception, right? Can we really treat that disorder when somebody's 10, 3, 20, or not, right? And so there are ways of turning genes on and off in mice. And so people are doing, throughout the field, they're doing experiments where they're saying, okay, we know this gene being off is bad. What happens if we turn it back on when the mouse is an adult or an adolescent or whatever you want to use for your mice? And strikingly, time after time after time, it's been shown in mice that when you turn the gene back on, even in adulthood of the mouse, you get recovery. Is it perfect? Not always, right? And would it be better to do younger rather than older? No question. But for every example that has been looked at carefully, turning the gene back on, in adulthood even, provides major benefits in terms of mouse behavior. Not the same as a human being, but still all we can do so far. And we also know that things like behavioral therapies, behavioral interventions, you don't have to, they work great when you're 2 and 3 and 4, but they also work when you're 15 or 16 or 17, right? So, the brain is basically plastic, can change. All right. And then the top genes are, quote, fairly common. In other words, they're not so ultra rare that there's no path forward for a company to kind of develop a drug there. But common in quotes, because 1% of autism or 1% of autism and intellectual disability is still 1 in 10,000, right? So they're rare, but they're not vanishingly rare. They get the benefit of orphan designation, but there's enough of a population for this first pass to actually, and I'm not talking from any knowledge, but just when I hear rumors, to actually develop kind of a plan that actually can pay for itself at the end. And as prices go down, as technology gets better, we can go lower, we can go deeper and deeper into that list and still have a good return on investment. Look at me using words I don't understand. So that's why I think right now, I hope that's why biotech and pharma are beginning to pay attention, because there is a way forward. And any drug that works for any rare disorder of this kind will, of course, immediately be tried in a broader group of people, right? And we'll be able to, if we have a drug that works, people will make the effort of finding in idiopathic autism or idiopathic intellectual disability, is there a subset of individuals that we can use that drug for that will work? In other words, can we step away from genetics and use other markers to actually put people into classifications to get a drug that works in one group, show that it works in a larger group? All right, and they're highly penetrant. All that means is that if the mutation is there, they explain much of the risk, right? So if you fix that, you're changing the whole risk profile for that individual. And I'm getting some signs that my time is up. Those are all the points I made, but now with data, but we'll skip over that. I'll just show you 2 slides quickly. This one over here. So this is one of the things that we study a lot at the Seaver Autism Center. It's called Phelan Mcdermid syndrome. And we've looked at deletions and point mutations. But the key point is here that these kids, especially with the deletions, have profound, severe intellectual disability, right? They may have no language frequently no language. These are very severely affected individuals. And as I said, there's a huge unmet need, and there's, to be frank, a lot of room for improvement, right? So we don't need a cure, we need a treatment, right? We need things to be better. Someday they'll be perfect, but there's a lot of room to just improving things. And we can talk about some of the life history of these individuals, and you'll all be weeping, right? So there's plenty of places we can make a change in these individuals' lives without saying we have to cure it today. And this is some of those experiments that show that when you turn a gene back on, in this case SHANK3, in mice, a lot of recovery happens even in adulthood. But it's mice. Okay. And this is, maybe I'll keep on talking even though I got my third warning. So I mentioned DDX3X, one of the genes we study, you'll hear about later. It explains 1% to 3% of intellectual disability and development delay in girls. This is fairly big. You've heard of Rett syndrome. This is much bigger. SHANK3, we could say it's 0.5% to 2%, depending on who you ask. I say 1% of both. That's 2% of 2%. That's not a trivial number of individuals. ADMP, some people are saying it's about 1%. Certainly some of the figures I showed you, ADMP was at the very top. That's kind of like 1% there, and so on and so forth. So there are lots of these genes. They're highly penetrant. I'll skip that. And so, all these things are true for these genes, right? And so a while back, I was at Takeda, and I told them about a new syndrome. And I said, I'm not going to tell you what it is, but I'm going to tell you what we know about this syndrome. What we know is that the cause, I'm ruining the punchline, that the cause is unambiguous and uncontested. We know the cause of this Mazuyev syndrome. Who speaks Hebrew here? There's an easy and accurate test for this syndrome. There's very high conversion. If you're positive on the test, you're going to get the syndrome. There are cell and animal models that can be used in drug discovery and development that are very, very relevant to the disease. There's good preclinical evidence that if you reverse the mutation, in this case, you get recovery in animals. The prevalence is not high, but not low either, 1 in 15,000. Lots of engaged families with plenty of registry information. Families want to participate. There are some expert sites out there that are actually learning about the syndrome and can advise you on endpoints in clinical trials. And the FDA is very supportive because this rare disorder is so devastating, they want to help. Okay? And then, as I said before, if the drug is successful in Mazuyev syndrome, you may be able to pivot and use it for other syndromes. So I asked the audience, I said, would this be something you'd go after? And they all nodded and said yes. And then I showed them the exact data that supports most of these things for one of the disorders we study, and then they began, and apologies to anybody from that group in the room today, to say, oh, yes, but, right? And I said, wait a minute, let's not go yes, but. You tell me that if you had all of these things, there's a path forward. Let's just focus on that part, because every one of these things is actually true for Phelan-McDermid syndrome and some of the other syndromes I mentioned. But of course, it's nuanced, right? Some people argue about the mouse model being perfect. I get that. But to a first approximation, this is true for all the top genes I've mentioned. And it's going to be true probably for the next tier as well. We just haven't studied them as much. So there's not anything called Mazuyev syndrome. Mazuyev is Hebrew for fake. There's no such thing. But there are probably 100 syndromes just like this that you could check off most of these boxes. And we can argue about the nuances. Yeah, is it a big check or is it a small check? Is it a check with a question mark? I get that. As I said, we have 3 constituents here. We have the families at the cutting edge or the bleeding edge of life with these disorders. We have people like me who are scientists who, ivory tower folk barely understand the real world. And we have industry here who's going to think about it in a very concrete and targeted way and say, is this feasible? We can't drive our company to extinction by going after chasing rainbows. What is feasible today? So I'm going to present my side, which is, these are all pretty much true, right? And pharma will come and say, how do we get a little further to convince ourselves? And families, who are all here today too, should turn to your pharma partners and say, how do we get you to a point where you are convinced that Joe's Mazuyev syndrome applies to my gene disorder? So I think I'll stop there. In the sake of, and thank everybody. This is our annual meeting. This annual conference within 2 business days is followed or preceded by our site visit, where the Seaver Foundation and the advisory board come together and give us really good advice and strong critiques, hopefully not too often, but certainly when necessary. They're not a bashful crowd. So I want to thank all of you for attending, and again, the 3 constituents, the Seaver Center, the Seaver Foundation, our board, and I'll stop there.